Thank you for joining us for another biology video. Today's topic is enzymes and activation energy. The first question we need to ask ourselves is, what is energy? Energy is the ability to cause matter to move or change, and all life processes are driven by energy. Where does it come from? What is the source of energy? Well, ultimately, it's the sun. Remember that producers or photosynthetic organisms can take energy from the sun and convert it into chemical energy, which the rest of us use. Whether we're talking about photosynthesis or other chemical conversions, we know that energy can be stored or released by chemical reactions. And a chemical reaction simply occurs when bonds are broken and reformed to make different substances. When we write a chemical reaction, we start with the reactants on the left, followed by an arrow, and the products are on the right. What are the reactants and products for the reaction shown below? In this example, the reactants are glucose and oxygen, and the products are carbon dioxide and water. Some of you may recognize this reaction as cellular respiration. A term you may have heard when dealing with chemical reactions is metabolism. What does the term metabolism mean to you? Technically, metabolism is the sum of all chemical reactions in your body, the totality, all of your chemical reactions. So this includes not only breaking down foods for energy, but also building new compounds to make you, building your new structures. So literally, you are what you eat. When you take in protein, you break down that protein into its subunits, amino acids, and then rebuild those amino acids into your muscles and hair and nails. Since energy is released and stored in the breaking and forming of bonds, another important term to be familiar with is the term ATP, or adenosine triphosphate. This is the energy that our cells are able to use. What do you need? To make a boulder roll down a hill? To ignite logs on a campfire? Or to start a chemical reaction? That's right, energy. Your body needs energy to carry out chemical reactions too. And the activation energy describes the energy needed to start a chemical reaction. Even if ultimately a reaction is going to release energy, it still needs an input of energy to get the reaction going. Just like even though that boulder is going to roll down the hill, it still needs a nudge or a push to really get it going. Here we see a graph that represents activation energy. It shows starting with reactants, ending with products, and it says that in order to convert from reactants to products, in order to make this chemical reaction go, there is a nudge required. This is a slight push to really get that reaction to take off. This is an energy investment. It takes an input of a little energy to get a whole lot more out of it. It's kind of like the stock market. You got to put a little money in to get something out. So how do you think you could speed up this reaction? Well, if you think of it as having to climb up a little bit of a hill to get to roll down it, wouldn't it go faster if that hill wasn't so tall? And that's exactly what we can see. The reaction will go faster without needing as much activation energy. So there's substances called catalysts that can help lower this activation energy, and catalysts do speed up the rate of chemical reactions by lowering the activation energy. So we know that a catalyst speeds up a chemical reaction, but what is a catalyst? Well, a catalyst is any substance that lowers the activation energy, and they can be organic or inorganic. They can contain C's and H's or not. The organic catalysts are specifically called enzymes, and these are usually proteins. So enzymes are substances that speed up the rate of chemical reactions by lowering the activation energy, making that hill not quite so steep. Here we see a graph comparing the hills. Again, we're looking at cellular respiration, We have glucose and oxygen as the reactants, and no matter what, they're going to proceed through a reaction and release carbon dioxide and water. But notice that without an enzyme, a great deal of energy is required. That activation energy is tremendous to get that reaction going. However, with the help of an enzyme, notice the activation energy is substantially less, which means that the reaction can proceed much more rapidly. When we say that an enzyme speeds up a chemical reaction, it's because that activation energy is lowered with the help of an enzyme. The next question is, why does this really matter to us? Well, ultimately, organisms need to maintain homeostasis, an ideal internal balance, and enzymes help a lot with maintaining homeostasis. How is this? Well, without enzymes, chemical reactions would not occur quickly enough to sustain life. You don't have days or weeks to wait for your food to break down. You need enzymes to help lower that activation energy so that your food can be broken apart. Keeping our chemical reactions going keeps us alive, so thank goodness for enzymes. How do enzymes actually work? Well, before we see that, we need to look at a couple more important vocabulary terms. We already know that the enzyme is a protein or organic catalyst that helps speed up chemical reactions by lowering the activation energy. The substance that the enzyme works on is called the substrate. And the substrate interacts with the enzyme at a very specific location, and this is called the active site. For example, if we have this pink blob representing the enzyme, and the substrates are these little blue circles, the substrates will interact with the enzyme at its active site. A different way to look at this is by seeing the substrates fitting into the active sites of the enzyme. Notice these are little grooves where the substrates can fit, and this forms a temporary enzyme-substrate complex. While this enzyme-substrate complex is intact, the enzyme reduces the activation energy of the reaction and allows the reaction to proceed more quickly. For example, here we can see the two substrates being bonded together and released as a product. While the substrates were changed, they were glued together to form this product. Notice that the enzyme remains unchanged. It's now available to interact with a fresh batch of substrates and to help speed up this reaction over and over again. To reiterate, while the reactants are converted to products, the enzyme is not changed by the reaction. If you noticed, the shape of the active site was very specific to the shape of the substrates. We say that enzymes work like locks and keys. Each key only opens one lock. You can't use a key and open every single house on your block. The shapes are very specific. And it's the same with enzymes. Each enzyme is only designed to work on specific substrates. This enzyme would definitely interact with this substrate, but the shape would not be right to interact with this one. So only certain enzymes do certain jobs. Here's another way to picture an enzyme-substrate interaction. First, notice we've got an enzyme called sucrase, and that ASE ending is a great clue that tells us we're looking at an enzyme, and it has available active sites. It has grooves ready to interact with a substrate. Here comes the substrate, sucrose. Notice that ose ending, so who is that? That's right, that's a carbohydrate. So we've got a disaccharide coming into the active site of the enzyme. The substrate and enzyme bind. We have the temporary enzyme-substrate complex. And while there, the enzyme helps lower the activation energy for a hydrolysis reaction, whereby the sucrose will be broken apart, the products are released. However, the enzyme is not changed by the reaction. Reactants to products, but the enzyme remains unaltered. The enzyme did, however, lower the activation energy for this reaction, allowing it to proceed much more quickly than it would have otherwise. We can use computer modeling to predict the shapes of these proteins, these enzymes, and to show the grooves or these active sites where the substrates are able to fit. This picture shows us a model of induced fit. When the substrate fits in, that enzyme-substrate complex is formed. So clearly, three-dimensional shape is extremely important when it comes to enzymes interacting with their substrates. The three-dimensional shape of a protein can be quite complex. It's brought about by several interactions, and ultimately what matters to us is that structure determines function. It is the structure of the protein, or enzyme, that allows that active site to be present, which enables the substrate to fit in that active site and the reactions to be sped up. When the enzyme structures form properly, and when that protein stays intact, things work wonderfully. But there are certain factors that can change an enzyme's shape. And if the shape is messed up, the enzyme cannot do its job properly. If the active site is altered in any way, if the substrate can't fit, the enzyme will not be able to speed up the chemical reaction. Some of the factors that can impact an enzyme shape are temperature, pH, and concentration. All enzymes have optimal temperatures. If an enzyme is in an environment that's too cold, molecules move too slowly, and the enzyme is not very likely to bump into its substrate. On the other hand, if a temperature is too high, it can actually break some of the bonds and cause the protein to misfold. Again, if its active site is not the right shape, it probably won't fit the right substrate. The pH of an enzyme's environment also can impact the enzyme's ability to catalyze a reaction. If the environment is too acidic or too basic, this can also disrupt bonds within that protein, and the enzyme can misfold. Finally, concentration. If the enzymes are more concentrated, if there are more of them in a given location, they're more likely to bump into their substrates, and this can help speed up reactions further. When we look at factors like high temperature or abnormally high or low pHs, when we talk about the bonds in the protein becoming disrupted, we can see this polypeptide chain, this protein, has unraveled. And this is called denaturation, the unraveling of a protein. If this protein required an active site to fit with a substrate, it's no longer present in the unraveled version. So when we say that a protein has been denatured, it means the enzyme can no longer work as a catalyst. When you lose the structure, you lose the function. In this model, we can see that the substrate should have fit perfectly with the active site of this enzyme, but because of extreme heat, the enzyme has unraveled, the active site has changed shape, and that substrate will no longer fit. Any reaction required is no longer going to proceed. The loss of structure due to denaturation means a loss of function. While certain factors like temperature and pH can lead to denaturation, which can affect the protein's ability to catalyze reactions, inhibition is another factor that can change an enzyme's ability to do its job. An inhibitor is a substance that prevents the substrate from gaining access to the active site. Competitive inhibitors physically block active sites, while non-competitive inhibitors can bind to an enzyme somewhere other than the active site, which can change the shape and prevent the enzyme from doing its job. With this model, we can see how the substrate should normally fit into the enzyme's active site, and in this instance, the enzyme would be able to catalyze the reaction. However, here we see that there's another substance in the way. If the substrate tries to bounce into the active site, the competitive inhibitor will prevent the reaction from being catalyzed. Over in the non-competitive inhibitor, we notice a substance has bound to the enzyme, but it's non-competitive because it's bound somewhere other than the active site. However, its presence has altered the shape of an enzyme, and the active site is no longer available for the substrate to fit. In either of these instances of inhibition, the reaction will not proceed as normal. In short, enzymes speed up reactions by lowering activation energy. They're specific for certain substrates and they can be impacted by temperature, pH, and inhibitors.